Welcome to Grovebio channel. In today's video, we're going to see about DNA replication in prokaryotes. This is the second part of the video which deals with the elongation. To know more about the initiation, click in the description below or the icon above. Now we can see about elongation. Elongation, the enzyme involved are DNA polymerase 3. This is very important enzyme, but unfortunately it can't add the first nucleotide. Why? Because it can only extend the nucleotide when previously 3-OH is present. Okay, when 3-OH is present, it can add the nucleotides along with it. But first nucleotide cannot be fixed by DNA polymerase 3. And it is in need of ATP. And next is RNA-H. This is indeed a very important enzyme because at the end, we don't need any RNA primers to be present in the newly synthesized strand. So, to remove the RNA primers, RNA-H is needed. And next is DNA polymerase 3. This is very special polymerase which no need any energy, that is, which no need any ATP for its action. And then it can proofread the DNA and it also has special capabilities 5' to 3' exonuclease activity, which means it can eliminate the nucleotides at the end because exo means end, and 3' to 5' polymerase activity, that is it can polymerase means it can add nucleotides. So DNA polymerase 1 is indeed the special enzyme. And next is DNA ligase. It seals the nick. Nick means a small groove or gap present between the nucleotides. At last, it is sealed by DNA ligase. Now we can go to elongation and how the procedure proceeds. This is the replication fork. To know the elongation, only one point is very important. The parental strand 3' to 5' is very important because from the 3' to 5' only the newly synthesized will proceed. Okay, so the newly synthesized will be 5' to 3'. Okay, either it is leading or lagging strand. The strand will proceed only from 3' to 5' of the parental strand. In leading strand, only one primer is present because it proceeds along the 3' to 5' direction and it proceeds along the helicase. And in 5' to 3', that is the lagging strand, here also the DNA synthesis can be proceeded only from the 3' to 5' of the parental strand. So there are many fragments of strand present there at the lagging strand. Now we can see how it proceeds. Here you can see the helicase which unwinds the DNA. So here you can see DNA polymerase 3 which fills the nucleotide after the RNA primers. As I said, it can only add the nucleotides. Okay. In lagging strand, there is more RNA primers and DNA polymerase fills the gap between the RNA primers. This is known as Okazaki fragments, named after the author Okazaki. So here in newly synthesized strand, the RNA is present, RNA primers is present. So in order to remove that, RNA-H is needed, and in order to fill the gap, DNA polymerase 1 is needed. So DNA polymerase 1 along with RNA-H comes. So here you can see gaps. There is 3 gaps at the leading and lagging strand, and we have to remove the RNA primers. So RNA-H and DNA polymerase 1 comes here and it moves along the strand, and RNA-H remove the RNA primers. So you can see the white strand is gone, and at that position red strand, that is DNA, is filled by DNA polymerase 1. But you can see gaps over there. To fill the gaps, what is needed? DNA ligase is needed. So there DNA ligase fixes the gaps. Thus the newly synthesized strand is formed. So here you can see DNA polymerase 3. As I said, in lagging strand, only one DNA polymerase 3 is used. Though there are many RNA primers, how it is possible? So here you can see replication fork. What happens? DNA polymerase 3 along with the beta clamp, it will be present. The DNA polymerase 3 has 10 subunits. gamma, delta, tau, psi, psi, delta prime, alpha, theta, epsilon and beta. Beta is the clamp. So this clamp is responsible for clamping the DNA polymerase 3 to the template. So where and all the primers is present, it will be moved from the template but not entirely moved from the template. Okay. So when the RNA primers ends, it again clamps to the template, so that only one DNA polymerase 3 is needed for entire lagging strand. So DNA polymerase 3 forms loops in lagging strand, and then it binds to the template by using the lock mechanism with the other DNA polymerase 3 in the parental strand. You can see from this picture, so that it will move from the template but it is not entirely detached from the template, so that only one DNA polymerase 3 is enough for whole template. In next video we can see about the termination. Thank you. Subscribe to the channel and please press the bell button below. If you have any doubts, please comment in the comment section below. Thank you so much for listening. Welcome to Grovebio.